Well, dear students, today we are going to discuss development in Drosophila. So before this is starting the development in Drosophila, let's know something about the very interesting facts about the fertilization in case of Drosophila that is commonly known as fruit fly. After mating, a Drosophila female typically stores 300 to 500 eggs in a specialized structure to which we used to call a sperm thecae and the seminal receptacles. And the supply of the sperm enables the females to fertilize eggs for up to 2 weeks without remating with the male fly. However, the sperms of this fruit fly are very long. Mature oocytes are individually fertilized in the uterus. Sperms are released from the storage organs and they enters in the eggs through a tube-like structure to which we used to call micropyle. Although eggs activation does not simply depends on the sperm entry, but before that the egg should be dehydrated. So the dehydration of the eggs before fertilization is compulsory. So let's just move towards the development in Drosophila. Like many other insects, early embryonic development in fruit fly, that is melanogaster, is very rapid and it is of sensation time. What is sensation type of cleavage? Sensatial means without any demarcation, without any cell membrane, the nucleus divides so rapidly just after the fertilization. One to two, two to three, three to four, and so on. And within 4 hours, the eggs becomes like a mulberry. That is, so many nuclei are filled in a cytoplasm. And naturally, the division of the cytoplasm will be unequal. They because moving here and there, they are being embedded in the eukaryote. So for this rapid division, 13 nuclear divisions occurs to complete the complete cleavage. And in the absence of cytokinesis, producing an embryo of some approximately having 6,000 nuclei in a common cytoplasm. Hence, I am calling it to be a mulberry. So at the interface of the nuclei 14, these nuclei are being packed. They are packed in individually cells, and this process is known as cellularization. So all the nuclei are being packed in the embryo, and this process is known as cellularization. Soon after the cellularization is completed, so the commencement of the gastrulation starts. What is gastrulation? That is formation of 3 germ layers. So let's move ahead. Here by this picture, you can see this is the egg of the Drosophila, and this tiny tube-like structure is the arrow you are seeing here. This is the micropyle, and this how the nuclear divisions occurs. So the first is the egg is being fertilized. So after the fertilization, it becomes 2 eggs and this nucleus, and the division starts after the 4 hours of the fertilization, and it divides so rapidly. It divides and divides and redivides, and finally these nuclei they arranged at the periphery of the egg, and that is known as cellularization, and this layer is known as cellular blastoderm, right? Now next, move to the next slide. Most insects they undergo superficial cleavage, where a large number of centrally located yolk contains cleavage to the cytoplasmic rim only. They touches to the rim of the cytoplasm, not enters up to the nucleus level. It remains suspended till the nucleus are not being divided. And this is known as syncytial. How the nuclei are being formed as cell-like structures due to the embedding of the division and redivision, and such type of division is known as syncytial, and the derm or the layer is being formed is known as syncytial blastoderm. You can see with the next slide, you can understand very well. These nuclei, they divide and redivide, and in between the nuclei, the microfilaments and microfibers are being formed. they are being attached with each other, and this is a kind of protein known as actin. This is the cells, they are being attached with each other, and this is known as actin. And due to the division and redivision, they are being embedded in the yolk, and this cellularization and a cellular blastoderm is formed. That is a syncytial, or you may call it the superficial type of division. So let's move to the next stage. So after the 14th division, the nuclear division, what happens? The stage of the gastrulation starts. Here in this image, you can see this is that is image A. The first furrow appears, that is the ventral furrow, in the center of the embryo, which becomes a mulberry-like structures, and it contains approximately 1,000 cells. And these 1,000 cells, they later on being pinched off from this embryo and embedded inside. And the upper surface, it then looks like a very smooth surface. And that is the starting formation of mesodermal cells. So just after it, a cephalic furrow is appears at the anterior region, that is the midgut imagination, and this furrow is known as cephalic furrow, that is the formation of the head. And in figure number 3, here you can see, similarly, just after the formation of the cephalic furrow, at the posterior end, the few cells are being aggregated, and they are being tried to embedded inside and try to touch the ventral furrow. And due to which the embryo becomes being curved. It is being curved because the anterior and posterior ends both are just, they are trying to touch the ventral furrow. Hence, the embryo is being curved, and this is the formation of germ band. Here you can see this is the procephalon area, that is the after the cephalic furrow, and then the posterior and these cells, they move inside, as a result of which this embryo being bent in one direction, and this is the formation of germ band. So after this, so many divisions occurs, and this is the starting of body segmentation. However, it's not so simple. Each and every body segment is being governed by the genes, that which part of the segment will form. And this is the formation of the germ band. Ectodermal cells on the surface of mesoderm. Let's move to the next slide. Yes, here you can see these ectodermal cells on the surface of mesoderm undergo convergence. These mesodermal cells, they undergo convergence and extension and migrate towards the ventral middle line of the germ band. Next, move to the next slide. In this slide, you can understand in very well direction, very well. So what happened? There's ectodermal cells. This is the anterior, this is the endoderm, this foregut, hindgut, and this is the posterior endoderm. These are the pole cells. In this direction, you can see this is the mesoderm, this is ectoderm, and this is the ventral furrow. So the ectodermal cells on the surface of the mesoderm undergoes convergence and extension, migrating towards the ventral midline to form the germ band. That is the formation of the germ band. This was a germ band. And collection of cells along the ventral midline, that includes all the cells that will form the trunk of the embryo. This is the formation of the trunk of the embryo. These cells are being moving. That is, archentron is being formed, and that is the organogenesis. While the germ band is in its extended position, several key morphogenetic process occurs, and this process is known as organogenesis. The very interesting thing is that neuroblasts differentiate from the neurogenic ectoderm. Here you can see, that the neurogenic ectoderm. And the nervous system is formed by the differentiation of ventral part of the cephalic regions. Hence, the nervous system in case of insects is not dorsal, it is always ventral. That is the reason that how the ventral nervous system is being formed. The body segments, after this, the body segments, several segments are being differentiated, and each and every body segment, by this diagram, you can see each and every body segment is defined to form a particular organ. The first three body segments, this is the cephalic region, the first three body segments, they form the thorax of the insect. T1, T2, and T3. T1 is responsible to form the legs of the fruit fly. T2 is responsible to form the legs and wings, whereas the third part, it is responsible to form the legs and the hapters, that is the balancing organs of the fruit fly. And rest, 8 segments, A1, A2, A6, and 7, 8, they are responsible to form the head of the fruit fly. So that is the formation up to the segments, that is the gastrulation in case of uh, fruit fly, that how it occurs, and how the gastrulation and cleavage, that is syncytial cleavage, occurs. Now we will discuss in the next lecture that what are these genes which responsible, that which body segment inform what organs. So that's all. Thank you.